So I started with this joke, which is sort of a joke, but there's something important here, right? And what is important is the fact that we have very different cultures. We have very different backgrounds. We have very different assumptions. How many people here come from physics, computer science, engineering? And how many people here come from biology, biomedicine? So it's, you know, it's half and half, maybe a little bit more people, more quantitative people. And you know, I'm a biologist, and I learned when I was doing my undergrad studies that it would be very difficult for me to be as quantitative as the physicists. But I also knew that if I didn't have an interest, if I didn't show an interest, that I would be cornered in descriptive biology. I would be cornered in stamp collection, basically, you know, taxonomy, systematics, things like that, and which of course was very important 200 years ago. But now biology more and more needs methods for quantification. So I think this is a very fruitful collaboration. We know it is because in the past 20 years, physicists have been flooding neuroscience departments. And I think the reason is because in physics, you know, actually Guillermo was my comrade during PhD, and we published stuff together that was important for us at the time. And I learned a lot from him and from Marcelo Magnasco and from other physicists with whom I've been interacting. In a way, this group of people invited here have a lot to do with these connections. But for that collaboration to be really fruitful, both sides need to be very tolerant, right? The biologists need to be tolerant because they may have to explain over and over again, you know, things on cell biology. What is DNA? What is RNA? What is a protein? What is the turnover of this particular molecule, and what does it mean? Because, and in my experience, what we find is that people will not learn the first time they see it or talk about it, or even the fifth time. It takes many instances of repetition until it becomes something that is part of your culture. At the same time, we need people in the quantitative fields to be very tolerant with the difficulties with quantification that people with biological background have. I know that most biologists, they will have a blank every time they see an equation, and they will just not think further, and that makes people with quantitative background irritated typically. So for this collaboration to be fruitful, we need to be tolerant, and we need to know that only by repeating the interaction many times we'll come up with a synthesis that is better than the point of view of one side or the other. So we'll talk a lot about sleep, and we'll talk a little bit, if we have time at the end, about dreaming. We'll talk less about dreaming if we talk about it at all, because we know much less about it. Okay, there's much less information, objective information about what's going on during dreaming. But I want to reach that point in the talk because we now have a lot of evidence that dreaming cannot be reduced to sleep, right? So the stuff that happens during dreaming is, if you see it in different ways, it's very different from the mere act of sleeping. But we know that dreaming typically happens during sleep. So what it is in sleep that allows dreaming to happen, and why is it that dreaming is something more than just sleep? This is something I would like to bring to you. And again, in this, I don't expect you guys to memorize the molecules that I'm going to present to you, or the particular areas of the brain in which we're doing the recordings. This is the goal here is to motivate questions, is to give you some information that you didn't have before that will make you conceive a question, something that could be asked in an experimental way or in a theoretical way, something you could model, Okay, so this is the intention here. I have a lot of slides, and I'm going to show a lot of information. Some of it very light, some of it not so much. And but the goal here is not that you learn all those things. It's impossible, but it's that this motivates your curiosity and say, oh, this is something I would like to know more about. I don't know if you notice, the scene here, it seems like a normal scene, but when you look at it carefully, you realize this woman has a bush coming out of her head. So this is this is a good representation of what dreams are. They are a mix of familiar things with some unlikely connections, some unlikely associations. And why is that so? And what is the function of that, if there is any function to that? This is something, hopefully we can reach this before lunch. We cannot talk about this, or I wouldn't talk about this, without starting with the work of Freud. In this book, The Interpretation of Dreams, he proposed several things that are tremendously influential and that became influential in different areas of knowledge, but that were then later discredited. Okay, and one thing that I want to do today is to present those ideas to you, and then to come back to those ideas at the end of the talk and try to give a contemporary translation of those statements. So in this book he says that dreams contain day residues, and meaning that dreams reflect somehow what happens during waking life. He said that desires are the motor of dreams, that you can explain dreams by knowing what people want, and that dreams are the royal road to the unconscious, that the dreams would be a privileged way to access information from the unconscious. However, since he did not give operational definitions to unconscious or to desires, those statements were largely considered to be non-scientific, to be poetry, not something that you could test, that could inspire a research program. And that has to do with the nature of the Freudian method to observe human behavior. Freud tried different things over the years, and eventually he developed a method that we call today the free association method. You let people stay in a very comfortable environment, with very monotonic, without strong stimulation of any sort. People lay down. Typically they don't face the analyst, and they just let their thoughts freely associate, and they talk about this. And dreams are particularly good for that, because it's something that people, they had in a very private manner. And then by doing that, Freud said that he could map the mind of the person and see where there were problems and areas in which the person wouldn't go, and so on. And he developed his whole contribution in psychoanalysis based on this kind of data. We don't tend to think of it as data because he was not recording it. He was not doing any quantitative, semantic, or structural analysis of what they were saying. But in fact, it was all based on observations. What is the problem? The problem is that early on, as soon as he started to publish papers and books based on this, people said, well, yeah, sure. We have Santiago Ramon and Cajal studying the shapes of neurons. We have Sherrington studying the physiology of neurons. And then we have this guy studying blah, 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 the words that people utter. And that was used against him early on to say, this is not scientific. We should not take this seriously. To make it worse, and you may think it's weird that I'm going to bring this here, but you cannot talk about dreams if you don't. What is worse is what is that people talk when they are in this situation? When you are in free association in an environment that is safe and you don't feel observed in a bad manner, you'll talk about the trivial things of life, but eventually most people, they end up talking about the things that matter, the things that really matter. Love, sex, and death. When you put those things together, it's so horrible, it's so heretical. People don't take it lightly. If any of you felt a little weird to see those images here, imagine 100 years ago in Vienna. Imagine Freud telling people that their kids had sexuality, that women had sexuality, right? And that problems could develop in terms of behavior that came from repression early on. And because of that, Freud was really submitted to trashing. I mean, he was considered to be non-scientific early on. This book came out in 1931. It's a whole book against psychoanalysis. In 1938, the Nazis invade Vienna, and then Freud has to leave to London, dies the next year. Okay? But until the 50s, you would find that in the US, in the medical school, psychoanalysis was very influential. MDs used to have psychoanalysis as part of their education. And then in the 50s, two discoveries changed that. The first one was the discovery of Rem sleep. So by watching children sleep, Azerinsky and Kleitman described a new state of sleep in which the brain was not quiescent with slow, large amplitude oscillations in the what we call today slow wave sleep, but was entering a very active state, which it's so active that the French call it the paradoxical sleep, because you look at the cortex and say this person must be awake, but the person is sleeping. And this is when, a few years later, William Dement and Kleitman published a paper showing that, in fact, this is the state when we dream. Now we know we don't dream exclusively during REM sleep, but most adults dream during REM sleep. If you wake them up, they will report a vivid dream. Okay? When this happened, a very interesting turn of arguments happened also, because people said, well, if there is something like REM sleep that it harbors dreams, then we should not study dreams, because dreams are so particular. People have their own, impressions, and maybe it means nothing. It's so bizarre anyway. We should study REM sleep. So the scientific thing to do was to study REM sleep. You could imagine that this same discovery could boost the Freudian program in a different world, but it didn't happen this way. Because in fact, the Freudians were getting farther and farther away from science. Then Lacan came and said that science is a social construct and we shouldn't pay attention to it anyway. And then, I'm simplifying a little bit, but in fact, this sort of, the fact that the psychoanalysis was expelled from science was seen as a good thing by psychoanalysts. They were free to have their own discipline, but it was totally independent. Another thing happened in the 50s, which was the discovery of antipsychotics. Okay? A very influential theory from the very beginning of psychiatry is that psychosis is some kind of dreaming. That when people have a psychotic attack, they're having a surge of dreaming while awake. And this was something that Freud believed, Kraepelin believed, Bleuler, all the founding fathers of psychiatry believed in that. Then in the 50s, people discovered that some drugs that block the dopaminergic receptor 2 can pretty much stop a psychotic attack with a lot of bad consequences for the patient, but for society it seemed like a great thing. You can, the guy is causing trouble, you just give him haloperidol and that's it. The guy is going to stop the trouble. And then this whole notion that you needed to delve into people's minds to understand what they were dreaming with and why, and then the whole thing became it started to seem nonsensical. And then the stance that the Freudians assumed was also non-scientific, so it was perfect. It was like Israel and Hamas. Both sides loved to disagree. And then it led to this famous statement by Popper. Popper said, he put psychoanalysis together with Marxism, homeopathy, and all sorts of metaphysical bodies of knowledge. He said, psychoanalysis is simply not testable. It's irrefutable. And then, therefore, we should not bring any contribution of psychoanalysis into science. Now, this lasted for many decades. It's interesting, around the same time, a little earlier, Konrad Lorenz, one of the founding fathers of animal behavior, he actually had a very different opinion. He said, well, what is considered scientific in psychology these days, back then in the late 40s, was experimental psychology, behaviorism, if you will. Okay? Very quantitative. And then he said, well, this is great, but they are going to hit a wall because they refuse knowledge. They don't want to open the brain to know what's going on there. They just want to deal with inputs and outputs. So this is sterile. And he predicted, interestingly enough, decades ahead, he predicted what happened, which is that neuroscience or neurobiology devoured experimental psychology. So now people are doing experimental psychology, but they don't call it experimental psychology. They call it neuroscience. They're using the same, the same theoretical tools, but they are now adding stuff to it. And then he said, if there's any psychology that will last and be important in the future, is what he called depth psychology, Freud and Jung. And Lawrence said that, early on, when nobody else could see it, and he said, well, in the end, depth psychology is the one that we will have to come to terms with, because these guys are making observations about human nature that are very serious, and we need to really understand that. Well, I didn't go into Jung because he writes in a much more flourished manner than Freud, right? So if Freud is not science, Jung could never be science. But in fact, if you take the concept of collective unconscious seriously, you're really talking about instincts. You're really talking about phylogenetically selected memories, right? And we know almost nothing about this, but now we know about epigenetic mechanisms, for example, that may explain why certain experiences emotional experiences, fear and so on, get to be carried on to new generations. And so I think that eventually Jung will be integrated into science, and it's probably more biological than Freud. But Freud writes in a very clear manner, Jung didn't, right? Freud did not say that we learn during sleep or that sleep favors learning. He did not say that, but he came very close to saying it, because he said that dreams have a day residue. Dreams reflect waking life. Now, if we learn during waking, and dreams reflect waking life, then almost as a necessity, we learn during sleep. He didn't say it so clearly. Jung did. Jung said the dream prepares the dreamer for the next day, right? But the first people to measure this, to actually go ahead and do an experiment instead of just an observation, were those two guys, Jenkins and Dallenby, in 1924, working in Illinois. What they did was a repetition of a classical experiment in psychology, the experiment developed by Ebbinghaus in the 19th century, which is the experiment that described quantitatively for the first time how we forget things. So Ebbinghaus discovered in the 19th century that if you teach syllables, nonsense syllables that do not exist in the language of the participants, if you teach them to subjects, and if you ask them over time how many of those syllables they remember, there is a very clear decay, an exponential decay, that is universal, let's say. You see this in humans, you see this in rats, you see this in bees, depends on how you do the experiments, but you always see a decay. We forget. This is the forgetting curve. Psychology 101, I don't know. Now, what they did here that was different is that they compared the same subjects, okay, learning nonsense syllables in English, and then it was college students, so students that would go to learn this material and then go to attend a class, and then he tested them, and they recovered the forgetting curve. And then they compared the data from the same subjects, except that they were exposed to the syllables, and then they were allowed to sleep, and then tested. And as you can see here, there's a major difference. The retention is much better. It stabilizes at a much higher level, 3 to 4 times more recall. You see the same subjects, participant MC and age. So you see that it's a comparison within subject, very strong. And this led to the notion that sleep helps learning by protecting memories from interference, by stopping some detrimental process that comes from being exposed to more stimulation. Okay? This theory lasted for many decades. But eventually, people started to realize that sleep does much more than a passive protection. It actually enhances some memories and weakens some other memories, and also mixes memories, restructures memories. It took many years for people to follow up on this experiment. There's a couple of reports that I could never find the actual paper in the 30s and 40s. And then in the 60s, the Americans and the French had a surge of interest in this kind of experiment, but they turned it around. they said, let's teach stuff to rats and rodents, and let's deprive them of sleep for many, many hours, and see how that affects their performance. So in the late 60s and early 70s, there's maybe 20 or 30 papers between France and the US saying that if you sleep deprive animals, they will not learn properly in a variety of tasks. So, and when I started studying those things in the mid-90s, when I started my PhD, this is when I began to be interested in this literature. There was a big controversy, and this controversy lasted through the 70s, 80s, and 90s. And the controversy was very simple. Well, it had many elements, but it would boil down to one thing. Are the animals performing worse after sleep deprivation because they could not consolidate the memory, which was the explanation that these people in France and the US wanted to give? Or is it because the animals are stressed? Because the way that sleep deprivation is affected is very stressful. In rats, for example, you put them in a platform. They're, every time they lose muscular tone to go to sleep, they fall in water. It's a platform surrounded by water. So they go back to the platform. And then if you do this for 1 day, 2 days, 3 days, it's torture. I mean, it's or not torture, maybe not torture. I don't. Waterboarding, right? It's something that is very bad to the animal. The animals gets very stressed. You see, major changes in stress hormones, and then how would you parsimoniously conclude that this is a problem related to sleep deprivation and not to stress? And this controversy lasted so much that some pioneers in the field, like William Fishbein, one of those early guys in the 60s, he quit the field. I met him about 10 years ago when he was coming back to the field, and he said, well, now that it's settled and people are not yelling at each other at meetings, I can come back and work on this. Because it was too acrimonious. People were really, really hating each other based on this. And then in 2003, there was a meeting of the Sleep Society, the American Sleep Society in Chicago, to celebrate the 50 years of the discovery of Rem sleep. I was in this meeting. And there was a special session, which was a confrontation, cowboy style, between two of the most vocal opponents of the memory-sleep relationship, and two of the most staunch defenders of that. Okay, and this meeting had 200 people in the audience, these four guys fighting, and then at some point, Stickold said, look, these are my data. What part of P smaller than 0.05 corrected for Bonferroni you don't understand? And then Siegel stood up and said, this is ridiculous. I will never come to a meeting like this again. These two guys left, and 202 people were clapping and saying, yes, this is the end of it. And that was the end of the controversy. So what was the experiment? What was the experiment that produced that effect? So this is the one that did it, and this is what happened, what existed before. So through the 80s and 90s, Carlisle Smith in Canada was producing data like this. So these are animals that are trained to find the hidden platform. They are in the aquatic maze. This is a tank of water that is opaque, so the rats cannot see. So they have to swim, otherwise they will drown. But there is a platform where they can stand, but they cannot see it. So they have to navigate and find it. And in the first day, they take about 30 seconds to find the platform. Okay, and then what they did here was to sleep deprive the animals. I think it was just REM sleep in this particular paper, in different windows of opportunity. So 1 to 4 hours after the training, or 5 to 8 hours after, or 9 to 12 hours after. And then the next day they were tested on the behavior, and you see that the animals in the 1 to 4 hour group and the animals in the 9 to 12 hours group, they got better. They are now finding the platform in 15 seconds or 20 seconds. So a big change in performance. But the animals that were sleep deprived 5 to 8 hours after learning, they actually show larger times, significantly larger times, so up to 40, 45 seconds. So this is the kind of evidence that produced so much fight. Sorry. So Carl Smith would say, well, this shows that it's not the stress, it's the sleep deprivation, because the other guys were as stressed, and they were stressed before and they were stressed after, but this particular period is important. We still don't know exactly why 5 to 8, but there's now plenty of evidence that stuff that happens during training, like for example, induction of immediate early genes, which I'm going to talk a lot about, will have effects on other genes 6 hours later, 12 hours later, 24 hours later. So it's this notion that the time after learning has some windows of opportunity that probably coincide with cascades of gene expression. So once you activate genes like CFOS and ZIF, I'm going to talk a lot about this soon, but once you activate those genes, then once they're translated, the proteins come back in the nucleus and produce and enhance the transcription of other genes. And these things have a lag, and this is probably what was going on here. But this was not convincing. This could not finish the controversy. And then Stickle published this paper here that sort of settled the issue. Okay, and it's so simple that I still, to this day, I don't understand why it took so long for people to figure it out. What he did here was, this was a task that involved perceptual learning. People were exposed to a screen with lots of, say, letters T. Okay, lots of T's, and the task was to find an L, one L. So you get, in the beginning, when you're first trained, you improve very little, but then when you come back the next day, there is a decent improvement, good 15 milliseconds, and then it keeps improving over time. Okay, even though the training was given once. So if you come there, 4 days later, there's more improvement here than here. So what was the control, the magic control? The magic control was deprive on the first night, let people sleep on the second and third nights, and then test. And this is the behavior that he recovered. So they were impaired, they did not learn, even though they were not stressed, had 2 full nights of sleep. So whatever happened in the first night was important. Whatever happened in the first night somehow damaged the memory, okay, when the person was kept awake. And when that happened, when you think about it, say, oh my God, people fought 30 years when they could just, delay the testing. But then again, a good idea is something that somebody else had ahead of you, right? This is something that people didn't think before, and this made a whole new moment for the field because people stopped arguing about whether the phenomenon happened or not, and started to discuss how does it happen, okay? And also started to map the full extension of what's happening, of the sort of sleep-dependent benefits that memory gets. I'm not going to go into the details, but now we have evidence that both sleep phases contribute to learning. So both slow-wave sleep and REM sleep contribute to learning. If you study human sleep, you'll see that this division is not between 2 phases. You have an introductory phase, N1, when we have a lot of images, when we're falling asleep and we sort of have a dream. Then N2, which has a lot of spindles. These oscillations, we'll talk about them. Then you have N3, which used to be N3 and 4, so which is what we call slow-wave sleep. So the brain, the cortex is just slowing down. The neurons are synchronizing and the waves become really big and slow. And then the last stage, which is REM. But for practical purposes, especially when we're talking about rodents, we can talk about slow-wave sleep and REM. Because the introduction, the entry into sleep is very fast and it's interesting, but it's not so interesting. If you ask Robert Stickle, what is each sleep phase doing, he will say slow-wave sleep is very good to consolidate declarative memories. Memories that are hippocampus dependent, memories of places, people, events, right? Episodic memories, declarative memories, stuff that you can teach other people very fast. If I tell you the name of my grandmother, Gininha, you didn't know it, now you know it. You'll forget it soon, but I mean, you have this information instantaneously. Very different from procedural memories, right? I cannot teach you how to ride a bike by telling you how to ride a bike. You have to try and fail many times until you acquire this skill. Of course, I can describe it. I can say, well, you move one leg, then you move the other leg. That may help, but that's not the same thing as a transfer of information, right? So you don't learn how to ride a bike or how to dive or how to do capoeira instantaneously, but you learn the name of any person instantaneously. And it's different systems doing it, and we can infer that with declarative memories, the fact that it's so fast also implies that you're changing very little the system. You're changing a few synapses. There's a lot that is already coded there, and all you're doing is linking, you already have the syllables. If I say my grandmother's name was Gininha, then you're getting the G, the Ni, and the Nya, and you're putting it together, and it's there, right? And then people will say that slow wave sleep is good for declarative memories, and REM sleep is good for procedural memories. I know a lot of people that disagree with that, and that show interactions between those systems. So I would say that the state of the art is that we're not sure. We're not sure. We know that slow wave sleep is very good for declarative. We know that REM is very good for emotional memories, probably procedural, but probably depends a lot on the specific training paradigm that you choose. More than we would like to think. Those generalizations are not warranted. Okay. Recently, so if people ask me 10 years ago, or 15 years ago, do flies sleep? I would say, of course not. They rest. They rest, but they don't sleep. Okay, that completely changed over 10 years, mostly due to the work of Paul Shaw. One could argue that the criteria for sleep in flies are too lax, are loose. Right? If you apply the same criteria to vertebrates, you're going to say that amphibians sleep, that reptiles sleep, and fish sleep. And this is a controversy. We know that reptiles do, but we are not sure about amphibians and fish because we have a lot of criteria to fulfill. And in the case of flies, it's mostly inactivity. Okay, the fact that they are inactive for many hours, the fact that it's circadian, so they are inactive not at any time, but they're inactive at specific long stretches of time. And the fact that there is a rebound, right? This is a very clear mark of sleep, that if you're sleep deprived, when you have the chance to sleep again, you will sleep more than normally. So you have a compensatory effect. This is a trademark of sleep, and this occurs in flies. Why are flies so interesting? Because of genetics. So you can know very well the genetic background of the animal, and you can manipulate that genetic background to generate different behaviors. In this particular paper, they manipulated the animals genetically to have a channel, an ion channel that opens in a specific part of the animal's nervous system. I shouldn't call it a brain, right? That makes, when those neurons are active, the fly goes to sleep. It's what they call sleep on demand or remote control. Okay, whatever name gets you into science. But I mean, what they actually do is to produce sleep by raising the temperature of the environment. Those channels are sensitive to that, and then the ions start to flow in the cells fire, and that makes the animals sleep. What is interesting here is that they were able to show that if you train the animal into some fly behavior, in this case it was courtship behavior, they go like this, and they want to mate, and then you can show that this is something that can be learned, you can measure it objectively, and what they showed is that if you train the animals and you put them to sleep forcefully, the learning lasts much longer. So it's not just the strength of the memory, but it's the duration of the memory. Okay, so that creates a whole new, it's very interesting, the kind of things you can do with that. Then we come to more, so I'm here with those 3 or 4 slides trying to map a little bit what we know about what sleep can do to learning. We know that sleep has something to do with creativity, right? Kekulé attributed the discovery of the benzenic ring to dreaming with the uruburu, the alchemic symbol that told him it's not a linear structure. He couldn't fit the carbons and the hydrogens in a linear structure, but then he realized he had the inspiration to come up with this. This is very famous. Less famous, but probably much more important, is the conception of the periodic table by Mendeleev, which he attributed to dreams. And then a case in physiology, Otto Lowy, he was trying to discover how, is there chemical transmission of information in the heart? And he had a feeling that yes, but how am I able to test that? So he had a dream about an experiment first he had a dream about an experiment that could solve it. And then he woke up and said, well, tomorrow I'll do it. And then the next morning he forgot about it. He was like, damn, I need to really, next time I dream about this, I should go to the lab immediately and do it. And then he had the dream. When you're really focused, again, desires, right? Remember Freud? Desires have something to do with your dreams. So he wanted to solve it, and then he had this, he conceived the experiment in which he would take the fluids from one heart and put it on another heart and see that if his heart was becoming bradycardic, the other one would also, and then he discovered the role of acetylcholine and so on. And he got the Nobel Prize for that. Now, the problem is, how do you capture this in the lab? Because this kind of mental process is not induction and it's not deduction. Okay, it's not something you learned from your contact with reality and one by one with objects. Not something you just deduce that makes sense. It's what people call abduction. This is something. It's insight. It's a leap of logical thoughts that leads to some solid ground. So how can you capture that? Because when it happens, it's very fast, and then it happened. It's there. The first people to bring this to the lab were researchers in Germany, in Lübeck, the group of Jan Born. Now he's in Tübingen. But that was in the early 2000s. And they used a psychological test called the number reduction test. And we'll do it here very, very briefly. Imagine I give you this string of numbers, okay? And I want you to produce a solution to that with using two rules. Don't try to add those numbers. It's not arithmetics, okay? It could be A, B, and C. The rules are the following. If the two symbols are the same, so one next to one will result in the same number, 1, okay? So 4 next to 4 will result in the 4, and 9 next to 9 will result in the 9. Now, if I have 1 and 4, then the rule is you get the third one. So now I get the 9. If I have 1 and 9, I get the 4. If I have 4 and 9, I get the 1. Is it clear for everybody? So if I have here 1 next to 4, what is the resulting number? 9. Now I compare 9 with the other 9. What do I get? 9. And then 9 and 1? Here? 9 and 1? 4. 4 and 1? 9. 9 and 1? 4. 4 and 1? 9. 9 and 9? 9. So they did that. So people learned this thing. Any of you can learn this thing. You learned already. And now if I give you many of those strings of the same size and ask you to produce the solutions, which is the last number, you will take about 10 seconds, typically, okay? So they asked people so some of the participants were instructed to go to sleep, come back and do it again, and some were instructed to stay awake, come back and do it again. The next day, those that stayed awake, 80% of them went from 10 seconds to 9.8 seconds. They had a very small improvement that was actually related to a motor improvement in hitting the keys. Okay, very small, use-dependent learning. Okay, now 20% of them had insights, and they said, you know what? I figured out that I don't need to solve the whole thing because the second number is always the same as the last number. So instead of taking 10 seconds, people started to take 2 seconds. But only 20% of the subjects did that. Now in the sleep group, 60% of the subjects did that. So they got the insight that the second number was the same as the last one. And this was the case by design, because those strings here, they had this mirror structure that makes the second number be the same as the last one. So there was some implicit instruction, some implicit structure here that was not available to the subjects, and yet 60% of those that slept had the insight. Many of them did not know why. Some did, but many of them did not know why, but they knew that, at least they knew that the second was the same as the last. Okay, so this was the first paper that said this, that sleep facilitates memory restructuring. We could call insight memory restructuring. Why? Because if you're doing some behavior that is more adaptive, okay, that you didn't know before, unless you believe in spirits or divine inspiration, this has to come from your previous ideas. Your new ideas must come from your old ideas, except that you're mixing them, you're restructuring your memory. And sleep has something to do with that restructuring. Somebody asked me about the rules of processing during sleep, right? What is different during sleep? The fact that you have so little of these neurotransmitters that enhance contrast in the response of cells is probably related to the promiscuity of sense of processing, of information processing during sleep that allows for this restructuring to happen. This is another paper by the group of Yamborn. Yamborn is probably the leading guy in sleep and memory in humans nowadays. He's been doing many discoveries, and this is a particularly interesting study, and it brings a cautionary note to all of us that teach. I believe all of us here teach at some level, right? Or if you're not teaching yet, you will, very likely. This experiment was like this. They trained people to recall locations of cards, so spatial learning, and also to do some finger tapping sequences, so motor learning, procedural learning, okay? But the experiment had a trick. So the subject would come to the lab, and they would say, well, you should learn this thing, okay? Fine, people are paid to do this in Germany, so they will do it at the right time, and you can very well control how the training happened. And then when they were leaving the lab, the experimenter would tell them in a very nonchalant way, almost, en passant, say, well, you know what? This thing that you just learned, you will not need to be tested on this tomorrow. We'll test you on something else. I think it was 2 days of interval. So the black bars represented the when people were expecting a test the next day, and the white one is when people were not expecting. Okay? What you see is that the learning was much better when people were expecting the test than when they were not expecting the test. And they controlled for, it's not that people studied the test in the interval, it's just the expectation they had about the future utility of that learning. And then this gray bar here is cases in which they had a series of questions, again, all disguised in the middle of the questionnaires, in a way that people wouldn't understand what was going on, but they had questions to probe whether people suspected they would be tested, even though they were told they wouldn't. And in those cases, when you separate those groups here, you see that they are also doing well. So it's not exactly whether they were told that it would be used or not, but it's whether they knew that this would be, that they would be tested on the material. This shows that they had many more spindles, those 10 to 12 hertz oscillations that occur in the beginning of slow wave and also at the transition to REM. They had more spindles in the expected than in the unexpected. And then there was a very strong, well, not very strong, but there was a strong correlation between spindle counts and learning of the spatial memory task, which was not present for the unexpected, in the case of the unexpected testing. So what is the cautionary note here? When you're teaching a class and you say, well, this and this and this will be subjected to testing, and this other part here is just for you to learn, but I'm not going to test, you're pretty much telling the brain, forget about this, it's not important. And we do it all the time. We do it all the time because we tend to think, well, it's good that I'm restricting so the students know what they really need to learn for the test, but they will be curious and they will know about other things. If you really want them to know about the other things, don't say anything. Say that everything is going to be tested. And since we're talking about this, there's another issue. Hal Pachler, a psychologist, has produced very good evidence, and Jeffrey Karpik, showing that tests repeated tests, cumulative tests, actually are pretty much the only thing you can do to make sure that people will remember in the future. You know, it's better to be subjected to a test than to study. That's the finding. Okay, so then if sleep is strengthening memories, is helping to I didn't show this, but then selective forgetting, restructuring memories, so doing all those complex operations on memories beyond just passive protection. How would you study that? How would you investigate the mechanisms underlying those changes, right? So we already started to talk about oscillations. So in order to measure oscillations, or even before you measure oscillations, if you want to measure the activity of a single neuron, how would you go about that? Of course, you can do it in two major flavors. You can be inside the cell, right? You can pinch the cell and either go inside or be in contact with the cell, in which case we say you have an intracellular recording. So you need a microscope, you need tissue that is either separated from a brain, or you need a very small brain that can sit in the microscope. So it's a reduced preparation. You do it, I wouldn't say you do it one by one, because some people do 12, 15 neurons at the same time with this technique. Very difficult. But you do a very, very slow collection of your samples. It's very involved. Many things can go wrong, and the behavior is compromised, right? You cannot have complex behavior at all. Even if you have an animal that can, a small mouse that can be in the microscope, you will have to be immobilized. And again, I mean, the soma of a neuron, do you guys have an idea of what is the size of the body of the neuron? One millimeter? 1 micron. How many microns? 20, 30, 50, depends on the neuron, right? So if your table moves 10 micrometers, your neuron will burst. It will die in front of your eyes, right? After 2 or 3 hours of looking for the neuron. So this is not very practical. You have a lot of information. You know when these neuron depolarizes, actually some people say you can do ensemble physiology because you can know what are the inputs, and there's this whole argument, but it's very limited in terms of behavior. The alternative is to do extracellular recordings, right? So you have an electrode, the electrode is nearby the bodies of neurons, but it's not inside them. So it would be equivalent to having a microphone here, right? I have this here, and if you all talk at the same time, most probably I will say I have Guillermo, right? And if I'm here in between, I'm probably going to say, well, sometimes it's Guillermo, sometimes it's Daniel, and because they speak at different times with different amplitudes, I may be able to sort that mathematically, right? And there are techniques in which you can, for example, twist 4 wires together, and if I had 4 microphones that are at different locations here, then I could mathematically determine with a lot of precision that in fact I have Guillermo here and Daniel there, right? I could even get more. I could get to the second layer here and get 4, 5, 6 neurons from this one tetrode, okay? Now, it's a different situation from the first case, because in the first case, I know for a fact that this is one neuron, that it has a specific behavior like that. In the other case, I have to make some assumptions, okay? And depending on the impedance, for example, if I have a very high impedance electrode, then I can make sure that I only get Guillermo. If I have a very low impedance, a very low resistance electrode, then I will get the collective behavior of everybody. So when we're recording using electroencephalographic techniques from the outside, I'm getting the behavior of hundreds of, tens of millions of neurons, and with much less specificity, right? So there's a trade-off here. Is everybody together on this? By the way, so people are already telling me that they want to visit the institute. We're thinking of organizing a visit over lunchtime on Wednesday or Thursday for the students. The faculty will talk separately. And then you may be able to see, depending on, I will see if we can schedule, so that you can see a recording like that you can actually see an intracellular recording and see an extracellular recording going on, That would be very nice. Have to arrange with the students to see if it will fit. Okay? Questions? Okay, now how would you tell the basic sleep-wake states of a rat? What would it take? What would it take to tell whether the animal was in waking, in slow-wave sleep, or in REM sleep? If he was a fly, I would say, here's asleep, this is wake. And I could publish in Science or Nature just by saying that. But, you know, the standards are different depending on the species and depending on many other things. So for mammals, we need to go further than that. Not much further, but a little further. Basically, what we need is a combination of a behavioral record. I need to know the posture. I need to know if this ear was flicking, if the tail is flicking, if the eyes are moving or not. I can get this with good video recording. I can get that with some electromyographic recording. I can get that with accelerometers. I can get that with a combination of techniques. And this is very important. Behavior is very important. But I also need some information from the inside. I need some information about what the brain is doing. It could be a screw in the skull to get some cortical responses, or it could be an electrode inside the brain, for example, inside the hippocampus. These are recordings from the hippocampus when the animal is very alert, exploring some object, and you see a very clear oscillation here, an oscillation that we call Theta Rhythm, which occurs between 4 and 10 hertz. It depends on the species and also depends on the animal, and depends on the behavior. Theta Rhythm occurs here, you see in the spectrogram. This is a plot of time, this is frequency, and the brightness is the power. And you see Theta Rhythm very clearly here, okay, around 7 hertz. Then the animal starts to be drowsy, and you have lower frequencies, so delta from 0 to 4. So there's a slowing down of the brain. Okay, and you see here slow wave sleep. You see the amplitude is much larger. These are the slow waves. This is a slow wave, okay? And in the cortex, it's very clear as well. And when the animal goes into REM sleep, it goes back into Theta Rhythm, but you can see here, it's Theta Rhythm that has a larger amplitude, and this is more steady. This is very choppy. The frequency keeps going up like this, okay? Which speaks to this notion of paradoxical sleep. So you have an electrophysiological signature of waking, but here the animal is in REM sleep, okay? So with these recordings, we can dissociate this behavior in two. If you now go closely to the behavior record, you'll see that you could actually tell slow wave from REM from the behavior. Something we tried to do with birds a good 10 years ago in a clandestine manner. So you can, but nobody will believe you. If you only have the behavior and say, well, I think this is REM sleep because the animal is doing like this, you know, the rats, they do a lot of twitches with the ears, but that's not convincing. People will not buy it. You need to have a electrophysiological record together with it. And even sometimes people will say they will not buy it unless you have the electromyographic recording. In the beginning, until people started to record with multiple electrodes on a regular basis, I would say 15 years ago, I mean, right? I mean, before that, you had a few people that could do it since the 60s, but it was so complicated and involved, and it looked so cumbersome, even the equipment. And now it's you can buy an internship and record as you go diving, to Fernando de Noronha. You can take your laptop and you can record a brain on the boat, but that's very recent. Dolphins, right? So using these kind of techniques, what did people do to understand memory processing during sleep? The first breakthrough was this paper in 1989, published by Konstantin Pavlidis and Jonathan Winson, the late Jonathan Winson at Rockefeller University. And what they did was really the first attempt to measure the Freudian day residue. Okay? What did they do? They went to the hippocampus of the animals, to the dorsal part of an area known as CA1. And this has pyramidal neurons that have characteristic place responses. How many people here heard about place cells? Okay, so what are those place cells? Those are cells that will show increased firing when animals traverse a specific part of the environment. If you change the environment, the neurons will remap. So if the neuron is responding here, and now I remove this wall, the neuron may not respond here anymore. Okay, so it's context specific. Okay, and those place cells, they have been, a staple of neurophysiology for 30, 40 years. It's a major thing in neuroscience. What they did was the following: they identified pairs of neurons that had non-overlapping fields. So a neuron that will respond only when the animal is here, and a neuron that will respond only when the animal is here. They found pairs of neurons that would have non-overlapping fields, and then they restricted the animal using a plexiglass cage to one of the fields. So what that produced was a differential experience for the neurons. This neuron was very experienced, Jimi Hendrix style. This neuron was not experienced at all. This guy was firing a lot for a long time, for 15 minutes, 30 minutes, I don't remember, and this guy not firing at all. Then they put the animals back in the home cage and recorded sleep. And what they got was this sort of plot. Okay, this is firing rate, spikes per second. If you compare post-exposure, meaning the time following exposure to the preferred field, to post-non-exposure, meaning the time following not being exposed, so the other neuron, basically this neuron versus that neuron, you see that during waking, there is a difference, but it's not significant. There's something there, but it's not significant. But when you reach slow wave and REM, there is a significant difference. Meaning, this neuron here that fires more during waking also fires more during sleep, even though the animal is in neither place field. So now we have a neuron that responds to a place field in the world, then during sleep, out of this place field, it remembers. Okay? Of course, firing rates are very, it's the most basic thing you can do with the data. But it's also perhaps the most solid. Very clear. Okay? Neurons are responding differentially during sleep according to the experience they had during waking, which is exactly the notion of day residue. There's a day residue here. Okay? This finding was taken to another level. And actually it's interesting because Jonathan Winson he had a very interesting history. He became a scientist when he was 45 years old. He was very rich. He came to New York and he said, I want to become a scientist. He offered his work for free at the university. He became a technician, then a researcher, then professor, then full professor. And he went to New York because he wanted to do psychoanalysis. And he did those experiments because he was Freudian. Now, back then, to say that would get him expelled from Rockefeller. So he wouldn't cite Freud in his papers. But I had the chance to know him, and he would say, well, my inspirations for these kind of experiments were the day residue. So in that sense, we can start to see that Popper was wrong, that psychoanalytical contributions are not all untestable. At least some are testable in a very reduced manner. Then this notion that experience-dependent changes occur at the level of firing rates was pushed to another level. In 1994, a very famous paper was published in Science by Matt Wilson and Bruce McNaughton. And this is an image that most neuroscience students know by heart. This is a very famous image. And here they were dealing no longer with firing rates, but they were dealing with relationships. They were dealing with how different neurons synchronize or anti-synchronize to each other. Okay? And what you see here are each dot represents one neuron from the hippocampus of a rat. And each of those lines represents the correlation, the linear correlation of the activity of those neurons, either positive or negative. Green is positive and red is negative, I believe. Okay? This is sleep before running a maze. This is running the maze, a very simple maze, circular maze. And this is sleep after running the maze. This is all slow wave sleep. Not because it doesn't occur in REM, but because REM is so scarce. The amounts of REM are are very reduced, so people tend to focus in slow wave sleep. What you see here is that before the training, there are many correlations between neurons, but they are very weak correlations. During the training, most of those disappear and other correlations appear and they're very strong. And after running the maze, you see that it's more messy, new correlations come up, but then some of those correlations that occur during waking, like this triangle here, they are reenacted here, right? So this has the same flavor as the Pavlidis and Winson finding, but here it's perhaps more useful for a theory of representation. Nobody believes that neurons are representing things in the environment just by changing their rate. Nobody has this theory, or nobody had this theory for decades now. But we do believe, most people believe, that representations are stored as relationships among neurons, right? And this is relevant because those changes in firing rates, they tend to be very short-lived in the hippocampus. Typically less than 20 minutes. If you track the sleep of the animal after learning for more than 20 minutes, those changes will just decay and go to nothing. Okay? In the cortex, it's a different story, and I will tell you about this soon. But in the hippocampus, it's very short-lived. So if you had to store representations with changes in firing rate, your head would explode. How would you keep all your memories if you have to increase firing rates and keep them high? That would not be feasible. So when they show that you can perhaps store them as relationships, then you can still have the same rates, but now you have different relationships. The neurons don't talk more, but they talk differently to different neurons. And that's a very compelling idea, and it's something that has stuck to this day. Okay? Many other papers came out of this. Typically, people don't even know about this paper. They only know about that one, but they mentioned now, so many examples of that in rats, in mice, in monkeys, okay? In humans, it's more complicated. We have examples of that using different techniques, but there's no paper that I'm aware of looking for neuronal responses in humans and sleep-dependent learning, typically because you have to record from a person that is undergoing epilepsy surgery or something like that. You cannot record for hours. You have 20 minutes to do your thing, okay? But there's one example from the birds. I was saying the birds are so different, but for some things they're tremendously useful, okay? And so there's one example that for me is the most compelling. You can notice here in the rats that this replay, if you call it a replay, they did, okay? If you call this a replay, you have to admit it's a low fidelity replay. So there's a pattern that is repeated, but there's also noise, or something that is not the pattern. But there is one animal, one case in which we have very strong evidence that there is replay. And this is the study done by Dan Margolish in Chicago with the zebra finch. The zebra finch is an Australian bird. This is a male. The females don't have this color in the face. This animal, it's what we call the closed-ended learner. It learns the song from the father or tutor in the first 2 months of age or 3 months of age, and then it rehearses that, crystallizes a copy of that song, not a perfect copy, a little bit of change, but not a lot of change, and then it keeps the same song forever until it dies. The animal will sing hundreds of thousands of bouts of song over the years with exactly the same spectral composition, very little change. Unless you make some lesion to the brain, the song will not change. Okay, so it's very strong memory. This is a spectrogram of a song of a zebra finch. Each of those structures here is a syllable. Okay, this is frequency, this is time, and the brightness is the power of the vocalization. So they're not melodic. This is something like meh, 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 something awful for our ears, but the females love it. The females of zebra finches, they will select their mate based on the songs, and interestingly enough, and Freudianly enough, they will choose the males that sing more close to their fathers. They prefer songs that resemble the songs of their fathers. Okay, so if you stick an electrode of the father of the female, sorry, so the female prefers the song of the male that sings like her father. Yes, to put it very clearly. Which is scary, right? It's scary. Now, there is a place, so if you get a songbird, the songbirds have a general feature, which is the so-called song system, something that was discovered in the 80s, the 70s and 80s by Fernando Nottebohm, Konishi, and others. Okay, what is this system? It's an area on the top called HVC, the high vocal center. It used to have a different name, then they changed all the names and it kept the acronym but changed the name. So the HVC is a region that if lesioned, the animal will not sing. If stimulated, the animal will sing. So it's like the Broca area, in a way, okay? And it has an analogous well, forget about the anatomy in birds. Fine. HVC projects down to nucleus RA, which is a motor nucleus. When you lesion RA, the animal will not sing. But if you stimulate RA, the animal will not sing a full song. It will sing syllables. So RA is like a storage of syllables, but not of a full song. Then RA projects down to nuclei that control the syringe. So the vocal organ is controlled by these descending motor projections. If you put electrodes in RA and you record the animal when the animal is singing, you're going to have neurons that have particular signatures of activity. So for example, this neuron here will give 3 bursts to the first syllable, will not respond to the second one, will give 1 burst at the beginning of the 3rd syllable, 4 and then a 5th burst on the 4th syllable, will skip the microsyllable and will mark in the middle of the last syllable. And if the animal sings 100 times, the neuron will fire precisely at those times. You change your electrode, the position of the electrode, you get a different neuron, you get a different signature, but it's this reliable signature. Is that clear? Now what happens when the animal is sleeping? What happens to this neuron when the animal is sleeping? This is what happens. Now, this for me is very striking because again, I mean, we like math, we need math, we cannot live without the math. But when you have, what was the definition that you gave, Don? Something that is fucking obvious to see. FOT, fucking obvious to see. This is FOT, right? You have something, you look at it and you say, okay, you know, there was no data massage here, I did not, you know, use a Jacobian, whatever. This is what it is. You record and you get that, okay? And that's compelling. That convinces people that indeed in RA, during sleep, the neurons are replaying. In that case, I think it's warranted. The neurons are replaying in the exact sequence, sometimes with a little compression. There's a little, sometimes, you know, here they skipped this burst, they add this thing here, maybe this is this one dislocated. I mean, but it's very clear that there is a day residue, that there is an electrophysiological reverberation of something that the animal rehearsed during the day. And something the animal did not know from scratch. It was not born with this song. It had to learn the song. Now, why is it that the neurons in RA reverberate memory so well, and in the hippocampus, in the mammal, you don't see this clear-cut replay? It's more messy. And the answer is, in RA, all the neurons care about only one object in the world, which is the bird's own song. The neurons don't deal with anything else but the bird's own song. While in the mammalian hippocampus, each neuron is actually dealing with different things. The neurons are, the technical word that we use, the jargon is multiplexing information. They're dealing with lots of representations and they're participating in lots of representations. So of course, it's a very interesting case, but it also tells us that we can only do so much with the birds. If we want to understand what we do, we will have to study systems that are more complicated than that.
Yeah, the precision of the spikes, it's astonishing, right? It's millisecond or sub-millisecond. So just in terms of feedback, I mean, if you think as an engineer, you need all sorts of checkpoints and feedback in order to keep that precision, even if you're not learning. I mean, learning perhaps is not that difficult, just add some spike here and there, but keeping the exact timing of this is a very good, difficult problem. So that's one interpretation to me.
There's one paper by Offer Chernikovsky and Partha Mitra and other folks, Science 10 years ago, something like this. Very nice paper, in which what they do is they record all the songs uttered by the zebra finch from hatching to adulthood. Right? They don't miss a single one. They get everything, and then they plot the similarity. They use Wiener entropy and other things to probably know the work. So they measure the similarity to the template. So what they're measuring is what is the animal producing in comparison to the target song that he learned from his father, or in this case from a decoy tutor. What they see is something like this. The similarity during the days increases, okay? And then the next day, so this is the night, and then the next day it does like this, and then this is the night, and the next day it does like this. So every day when the animal wakes up in the morning, the song is worse than yesterday, okay? It's not as good a copy of the template as he was singing at the end of the day, the day before. But when it goes up during the day to a higher level, so it's 2 steps forward, 1 down. It goes like this: 2 down, 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 okay. And they say this is probably similar to simulated annealing. So it's looking for trying to find a global minimum instead of a local minimum. So you have to, the animal learns and then it has to unlearn a little bit so that it can learn more. Okay? Now what is very interesting, and then it has to do with your question, which may indicate that this is actually adaptive. The ones that make the back step larger, the ones that have a degradation of the song, a larger degradation of the song overnight, are the ones that reach the best imitation, which suggests that it's not so simple as rehearse and get it, but it's about how you. So it's about finding the many ways, and it's about muscle control. Many ways in which you can utter something that is really similar to that template. And why would that be important? Because that template comes from somebody that was able to produce offspring. So it's true and tested. It worked in the real world. Right? The animals know for a fact that their fathers were able to mate. Okay, fine. Now, you learned something, and you learned something so well that you reverberate it in your circuits. Your neurons are now, there's a pattern that was imprinted there. How do you keep it for years and decades to come? You cannot keep it, you cannot store memories in an active manner, right? If I ask you what was the name of your first pet, your first dog or cat, you remember. You just remembered because I asked you, okay? You were not thinking about that 2 minutes ago. So somehow you have a latent memory that we are able to retrieve, and then it becomes active, and then it involves changes in firing rates of neurons. But when it was latent 5 minutes ago, it was not about rates. It was about something else. That something else is what Donald Hebb called in 1949 morphological changes. So in the beginning you have reverberation of memory, but that reverberation leads to morphological changes, what we call today synaptic plasticity, neuronal plasticity, okay, circuit plasticity. So the memories, latent memories, are not stored in the activity of neurons in most cases, but they're stored as morphological relationships that lead to physiological relationships. Okay, if we had to give names and information about that currently, it would be a slide like that. I don't want you guys to remember this slide or the details. What is important here is glutamate will open channels, will allow for sodium to get in through AMPA receptors. Eventually, calcium will get in because there's a magnesium here that will leave, and then it will activate a cascade of events which involve protein kinases, so proteins that can phosphorylate other proteins. You don't need to know the details. The important thing is that there's a domino here that occurs in the first 2 seconds, and then after a few minutes, this domino will lead to the phosphorylation of, for example, Crab. CREB is a transcription factor. It will help the transcription of different kinds of genes. Some genes we call effectors. Why? Because once the RNA comes out of the nucleus, it will be translated. Actually, in this case, the translation is at the level of the dendrite. And the protein itself, ARC, in this particular case, will do things directly. So ARC will be translated in the dendrite and will be interacting with AMPA receptors, with actin, with CAM kinase 2, one very important kinase, and will do things to remodel the synapse. Right? So it's like a worker that goes there with his hands and does things on its own. Now these guys here, the regulators, they don't do that. Why? Because they code not for proteins that can work all in the metabolism or right away at the synapse. They code for transcription factors. They code for proteins that will return to the nucleus and then will work like CREB, will promote the transcription of other genes. So for example, ZIF268 has binding sites in the promoters of over 300 genes. So if you increase the expression of ZIF268, that's going to cause a change in the expression potentially of 300 other genes. So it's a big amplification of the signal. And that's also a reason why, that has to do with your question, those gene expression responses, they have windows, they have waves. You know, it takes half an hour to reach the peak of RNA for these guys, 1 to 2 hours to reach the protein peak. So that means 6 hours later, the consequences of that will be felt, okay? Because other genes will be transcribed in more or less amounts due to the activity of those transcription factors. Am I speaking Greek here? Are you guys following? How many people knew those things more or less before? Yeah, good. So, what is the important thing? These guys here, they are the first genes to have their expression changed after stimulation of the cell. Because they are the first ones to respond, we call them immediate early genes. Okay? The RNA tends to peak half an hour after the stimulation. The stimulation could be anything from electrical stimulation to growth factors. What is important is that without those genes, you cannot have long-lasting memory. Memory that is encoded at the level of the synapse, let's say the phosphorylation of calcium commodulin kinase. It's an enzyme that has 4 sites for phosphorylation, and it phosphorylates itself. So once it's phosphorylated, it's a memory unit, because it will keep itself phosphorylated for a long time. But that will not last for hours or days. That will last for seconds and minutes. Okay, maybe hours depending on the situation, but certainly not for your life. So if the memory does not reach the nucleus, if the changes do not reach the nucleus, the memory will not be long-lasting. If you block protein synthesis, if you block this process here, you block memory. You block long-lasting memory. Okay? And this is very important. Why? Because once you have these things expressed, and once the consequences of their expression have been made, then you can have, for example, the creation of new synapses. You can have the multiplication or the reduction in the number of receptors at the level of the synapses. AMPA receptors, for example, can be internalized or can be recruited in the case of silent synapses. So I know it's a lot of details, and I know, you know, we're not going to ask this, and you know, what is the sequence of kinases? No, not at all. But we want you to get the gist of this. That there are molecular changes that take seconds to minutes that will determine whether the memory will last hours to days to years. Okay? And then we come back to an issue, which is the environment that I encountered when I was starting my training in sleep and memory. So back in the 90s, we started to know that sleep and memory are indeed related. That's good, fine. So opportunity for us to do research. However, the available evidence, mostly from the group of Chiara Cirelli and Giulio Tononi, was that sleep decreases immediate early gene expression in the brain. CFOS, ZIF268, Jun, MYC, several other genes. Okay? And that led them to say that during waking, those genes which are related, which were thought to be related to long-term potentiation, to the enhancement of synaptic strength, those genes would be upregulated, and during sleep they would be downregulated. So sleep would be about synaptic downscaling or renormalization. So they have a whole theory, very strong theory, and actually dominant theory, that says that all the benefits that we have, the cognitive benefits of sleep, have to do with a down selection of synapses. So whatever is weak gets to be erased. You bring everybody down, but those that are strong synapses will stay strong enough so that in the end, the signal-to-noise ratio of your memories is better. And that's how we learn during sleep. Okay? This is the strongest theory, and it has a lot of experimental support. The original findings were things like that. This is the original paper. They were coming from the group of Pompeiano in Italy. This is done with in situ hybridization, so they're basically throwing radioactive RNA probes that are complementary to the RNA that they want to study. And this is animals that were killed after sleeping a lot, spontaneously. And this is animals that were killed after 6 hours of being awake, a forced wakefulness. So you see there's a strong expression of FOS, of one of those transcription factors, in animals that were awake, and very reduced expression during sleep. Okay? This was followed up by several papers showing very similar effects. And they were able to lesion the center of the brain that produces noradrenaline, called locus coeruleus. And when you do that, this is a Science paper, 96, you get it's almost as getting the animal, it's the same as sleep, you know, no expression of those genes. And this is a nucleus that is shut down during REM sleep, so it all made sense. Okay? This is a review that was published this year, okay, in which they summarize all the evidence for that. So evidence like this one showing that in sleep groups you have less expression of AMPA receptors. So AMPA receptors are actually critical for any of those changes to happen, because they are the first thing to open and allow sodium inside the synaptic, the postsynaptic terminals. So there's a decrease during sleep. They show that cortical evoked responses are decreased during sleep. They show that miniature excitatory postsynaptic currents are also, well, this is a small effect, but anyway, they show that they are decreased as well. And they show, morphological studies show a decrease in the number of new synaptic boutons, okay? I'm not gonna go into the detail. I have a lot of stuff to show you if I can until 12:30. But anyway, there's maybe 40 papers on this, mostly from the same group and dominating the literature. And then it led to models. So this is a model. They basically have a pretty detailed model of a neuron with all the conductances and everything. And they show that the average connection strength increases during waking. And when they shut down the system from other inputs, then it will just come down during sleep. And this will lead the firing rates to come up during waking, and the firing rates will come down during sleep. And the slow waves will also attenuate. So what all this is saying, that they have empirical evidence and they can model it, and it's basically saying that during waking, the system is dominated by synaptic potentiation. Long-term potentiation is this particular mechanism, but there are many ways in which you can achieve potentiation. It's all saying that learning is happening, and so encoding is happening, those genes are being expressed, the synapses are getting stronger, but then during sleep, the system needs to come down to normality, they created this hypothesis, the synaptic homeostasis hypothesis, which in my opinion, they just adapted an idea from Gina Torrigiano. They said, whatever comes up needs to come down. So, well, but it's true. I mean, and it makes sense, right? So during sleep, the system would be dominated by, they changed the story. In the beginning, they said it was just a passive decrease, downscaling, LTD, long-term depression. Then they said, well, in fact, if the synapse is too weak, it should be stronger. If the synapse is too strong, it should be weaker. So you should homeostatically compress the dynamic range of synaptic weights. And by doing that, you would save energy. You would allow the system, you reset the system for new learning, which makes a lot of sense. And I think this is all happening. Okay, so this is the theory that is called SHI, Synaptic Homeostasis Hypothesis. I'm not going to be able to finish everything I brought to you, but we'll see what we can do. When I was doing my PhD, uh, back in 95, 96, I started doing experiments like that, like this here, okay? But I was not doing sleep deprivation. I was not forcing the animals to do anything. I was just letting the animals enter spontaneously into whatever sleep state they wanted. And I was also separating slow wave sleep from REM sleep. They never did in those studies because REM is so rare, so they were just focusing on slow wave. In the beginning, I was getting the same results as they were getting. When I was doing the first experiments, Guillermo remembers it, I didn't know about them because there was no PubMed, Netscape was still to be born, and I didn't go often enough to the library to know that they had already published something against my hypothesis. My hypothesis was, if we are learning stuff during sleep, some of those genes should go up at some point, okay? And then I started to see that they wouldn't go up, they were going down. And then I found their papers and I said, oh damn, I just replicated their stuff and it's not even novel. I mean, I can't even publish this thing, because they already showed it. I just showed that it was the same for slow wave and REM. And then I started to read a paper by an Italian guy called Antonio Giuditta from the 70s. And Giuditta said, it's not enough to study slow wave and compare it to REM. We have to think of the system as a system that processes information the way that our gut processes food. So I don't want to compare the esophagus to the stomach, but I want to compare the esophagus to the stomach with and without food. This is the critical thing. This is the argument that Giuditta put. So I said, fine, I'm going to take that to face value, and I'm going to compare groups of animals that went through waking, slow wave, or REM. But in some cases, the animals will never have left the home cage. It's the monotonous, boring situation. And in the other cases, the animals will be exposed to a rat's Disneyland. Okay, basically cardboard boxes connected with PVC tubing and a lot of new, unknown Fruit Loops hidden. And we got, okay, and then what is the rationale? The rationale is you expose the animal to the objects or not, let a few hours to pass, 3 hours to pass, so that the gene expression caused by these experiences is down. And then we study them during waking, slow wave, or REM, allow them to enter a reference state. Once they reach the state, we wake them up and keep them awake for 30 minutes. Why? Because this is what it takes for the mRNA to peak. So by doing that, we normalized the experience across groups in the last half hour. What was really different was the reference state 30 minutes before. Is that clear? Kill the animal, freeze the brain, cut the brain, hybridize. What we found was this. In the non-stimulated group, okay, animals that never left the home cage, the expression of ZIF268, which you see here in red and yellow, was very strong in the cortex and in the hippocampus during waking. But then it decreased during slow sleep and it decreased during REM sleep and it stayed low during REM sleep. Okay, so this is fully concordant with Tononi and Cirelli's theory. However, when the animals did have an experience beforehand with novelty, the expression decreased during slow sleep, but then it was reinduced during REM sleep. There was a rebound, okay? Something that maybe Freud would call a molecular day residue, right? So this is interesting because this is a gene that is important for memory consolidation. If you knock this gene out in a mouse, it will learn everything on day 1 and it will remember nothing on day 2. So this is important for memory consolidation. And then this gene is reinduced in the cortex and in the hippocampus, specifically during REM sleep. This is very interesting. So I actually, when I had this result for the first time, I invited Cirelli to my poster. It was the first neuroscience meeting I went, 96. And she came to my poster and said, I don't believe this. And for 20 years, she didn't believe it. And for 20 years, they never cited our papers. Never. But then, I'm going to show you, we added papers to this, and other people joined, and now there's this review that I showed in Neuron was the first time they cited us. One criticism that I got from this was that the experience was non-specific. Specific is a magic word in biology. You need to use it. If you don't use the word specific in the paper, you will probably not get through. So it was not specific. The animals were having motor experience, somatosensory experience. It was dark, so it was not visual, but they had olfactory experience. They were running around. So you have a reinduction of the gene throughout the cortex, in the hippocampus. It gets the amygdala. It's a mess. So we were compelled by the reviewers of those papers to say, let's try a reduced preparation. Let's do something more reductionistic, but that can be a little bit more specific. So instead of exposing the animals to that, exposing them to a novel environment with novel objects, what we did was to put an electrode in the perforant path in the axons that leads to the hippocampus to pass currents so that we could generate a response in the dentate gyrus, the entry point of the hippocampus, so as to produce long-term potentiation. So that mainly, it's like this. How many people here know what LTP is? Okay. The axons are coming to the dentate gyrus. The neurons here are receiving this information. You put an electrode here, and you put a recording electrode here. And to make it very simple and quick, imagine that in the beginning, I give a stimulation here, and I get a response here. Then I give high-frequency stimulation, okay? And then I get a response that is either bigger, or I get a response that is multiple. Now when I come back to one pulse, I still get multiple, or I still get bigger. So there is an enhancement of the response. I'm being very simplistic here, but there's an enhancement of the response to a single pulse of stimulation as a consequence of having had high-frequency stimulation. Okay. So by doing that, we could input an artificial memory in the dentate gyrus, something we could measure over hours and be sure that it was there, and we did. And then we said, so will that cause gene expression in the hippocampus, and what will happen? Well, we knew that would happen, but what will happen during sleep? And I'm going to show you the summary of the experiment. I'm going to delve into details later. Group data, this is individual animal data, just examples. I'm going to show you group data later. I'm not showing any slow wave sleep here, because slow wave sleep is always a signal to stop the transcription of this gene, ZIF268. Okay? I'm showing you a summary. So at 9 a.m., the animal was awake, and we induced LTP in one hemisphere. The other hemisphere was used as a control. It received no stimulation, or in some cases, low-frequency stimulation that did not lead to LTP, and it was the same as a non-stimulated control. At 9:30, one hemisphere, the stimulated hemisphere, showed very clear expression of the gene in the dentate gyrus, and nothing on the other hemisphere. Okay, so that worked. We knew it had to be like that. I mean, that's what we planned. Then comes the novelty. At noon, you let the animal stay awake for hours, and then you let the animal go to sleep. Then, when you kill the animals, you see no expression in the hippocampus and a lot of expression in the cortex and the amygdala. Less in the other hemisphere because they're commissures and connections, but this is not significant. If you let the animal wake up and have a second REM sleep episode, the hippocampus is still low expression and symmetric expression, but it's very strong lateralized expression in the cortex, in the somatosensory cortex, even in the motor cortex. So here we are 1 synapse away, here we are 3, 4 synapses away, here we are 5, 6, 7 synapses away. So apparently something is moving away from the hippocampus as sleep recurs. This is group data. I'm going to show you, this is all averages of all animals. This is a circuit that begins with dentate gyrus, CA1 fields, entorhinal cortex, auditory cortex, lateral dorsal nucleus of the amygdala, S1 and M1. Okay, so this is a rough sequence. This is what the reduced preparation allowed us to do, is we have an entry point for the memory, an artificial memory. What we found is this. In the beginning, you have gene expression only in the dentate gyrus, and very little. You see, this is close to 1. It's a ratio between successive states. So this is actually what happens. It's the ratio between after stimulation and before stimulation. So it's mostly in the beginning in the dentate gyrus. Then it moves to the intermediate regions of the circuit. During slow wave sleep, there's a shutdown. During REM sleep, everything comes up again, but reaching the intermediate levels. During late wake, it reaches the further levels. During late slow wave, it's a shutdown again. And during late REM, you reach further regions and not in the hippocampus. So this is showing that at the group data level, the gene expression seems to be displaced over time. Okay? So we interpret this as 3 waves, hippocampofugal waves of gene expression that were stopped by slow wave sleep. Slow wave sleep is the stop signal for those waves. Then we said, okay, interesting. Could it be that the expression that occurs in this intermediary region of the circuits, entorhinal cortex and S1, depends on the hippocampus. The hippocampus seems to be, being disengaged, but could it be that it's actually playing some role there? And then we did this following experiment. We induced LTP on both hemispheres, and we injected a sodium channel blocker in one hemisphere when the animal entered REM sleep. So when the animal started to go into REM sleep, you see here the very early beginning of theta rhythm. We kill with tetracaine, then you kill the activity. And what we found is that in the REM animals, the control hemispheres would show good gene expression, and the inactivated would have no gene expression. And if you did that during waking, you wouldn't have the same effect. Why? During waking, most of the cortical expression has to do with interactions with the thalamus, interactions of the ascending sensory pathways. During sleep, this is not there, so it depends on the hippocampus, and when you knock the hippocampus down, you affect the cortex remotely. So that says that those hippocampofugal waves, right? So what this is saying is that if you block the hippocampus at this time, even though it's not showing gene expression, it will stop gene expression remotely. And that made us think of a famous patient called patient HM. Who knows about this guy here? Okay, so this is the patient that was critical for our understanding of the role of the hippocampus in the encoding of episodic memories. This patient had epilepsy, the foci were the hippocampi were removed, he was cured of epilepsy and became amnesic for the rest of his life. Both anterograde amnesia, so he was never able to learn new declarative memories, but also retrograde amnesia. He forgot all the recent memories, he forgot all the circumstances that led him to the surgery, but he did not forget childhood stuff. So that was the first clear case. Well, there were cases before. Hebb was studying this since the 40s, early 40s, but this was a clear case that said the hippocampus is important for the acquisition of the declarative memories. It's important for the coding of those memories over time, but then if you wait long enough, you can do without it. It's not that it's not involved. Recent work shows that it is involved, even when it's long-term memory. But you can do without it. Okay, so it's corticalized. I did another experiment some years later in which I came back to the more naturalistic approach, so it's objects again, exposing the animals to objects. And what I did here was look for both ZIF and ARC expression in S1, somatosensory cortex, V1, visual cortex, and hippocampus. Everything done in the dark, so the V1 should, if it was textbook-like, it should be all negative. But the question here is, let the animals sleep freely for 4 hours, in average 28 sleep cycles, 28 full cycles, freely, and then you kill the animals. What do you see? So if you kill the animals right during waking with no exposure to anything, the gene expression levels are very low. If you kill them right after exposure to the novel objects, very strong expression, mostly in the somatosensory cortex, but some in the visual cortex, even though it's in the dark. And then also mostly in S1 cortex and much less in the visual cortex and the hippocampus. Now wait 4 hours and kill the animal in slow wave sleep. Nothing. Kill the animal in intermediate sleep, which is a 5 to 10 seconds transition between slow wave and REM. Nothing. Of course, I only took the trouble of doing that because I believed it was this guy here, because it's spindle rich, but it isn't. And then REM sleep. And then you have very clearly the expression in the cortex and supragranular, infragranular layers, nothing in the hippocampus. This seems like expression, but if you compare it to the control, it's not significant. So in fact, in a naturalistic situation, when you just let the animal sleep many cycles, you see that the expression is corticalized, it's not hippocampal. So brings us to this notion that, maybe memories are corticalized due to sleep or in relation to sleep, the sleep is helping that. Okay? Well, I sort of gave you half the story. Let me just finish the story with a little bit more, okay? A little bit more. Just 5 minutes more, okay, Guilherme? Okay. No, so this is I put a quote of this recent review in which they, for the first time in 20 years, they mention our papers. And they mention our papers. Then there are some molecular studies in rats indicating blah, 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 our papers. And then they said, well, however, ZIF and ARC are probably not LTP. They are probably LTD. So they turned their argument around. They use those genes to say these genes are like LTP and they go down during sleep. hence the hypothesis. Now that they acknowledge that those genes go up, they say, well, but there's evidence saying that it's more complicated and that in fact they probably mean LTD. Of course, this is tricky because I could go back to their original data set, so fine, so the reduction of those genes should mean LTP. And this would become an endless fight, okay? There's another way of going about this. The other way is to say, let's look for some marker of LTP that is not disputed. Okay, something that everybody agrees means LTP. And that could be the phosphorylation of CAM kinase 2. This is an enzyme, I mentioned it here some time ago, that will be active at the postsynaptic terminal, and it will interact with NMDA and AMPA receptors to strengthen synapses. Okay? And this is pretty much, everybody agrees that what it does when it's phosphorylated is to strengthen the synaptic connections locally. So we decided to chase that. We started years ago, and now we're trying to publish it. This is work done by Katja, Ani, and Vinicius. They were both my postdocs, and Ani is my students currently. Basically, we repeated the same design as before. Okay, the control animals never were exposed to any novelty. The exposed animals were exposed to those objects, were kept. Here it says 3 hours, but in fact, it was only 1 hour. This is wrong. It's only 1 hour. And then we let them go into the different states and kill the animals. So we repeated the same design, except that we now did not wait half an hour for mRNA of immediate genes to peak. We killed the animals immediately after REM sleep or immediately after slow wave sleep. Why? Because the evidence that is out there from Lisman and others is that CAM kinase 2 is phosphorylated within 2 seconds of stimulation. Okay? So we wanted to kill the animals immediately after the reference state and see what would happen. This is the labeling that you get for phosphorylated CAM kinase 2 alpha. So it labels bodies, it labels cytoplasm. So it labels cell bodies and it labels neuropil, it labels dendrites. Okay, so this is from CA1. And this is the labeling that you get for ZIF268. It's nuclear. It only labels the nucleus because the protein is localized to the nucleus. Okay, this is to show that when you do a Western blot, that you get the bands in the right place, that the antibody is good. And these are the data. We are currently trying to publish this thing. We had a 6 months interaction in Nature Neuroscience, which in the end did not result. And there was they sent it. All the editors liked it, and then the people that work there, they couldn't see a problem with that. They asked for more data, we gave more data, and then they sent it to the leading, quote unquote, the leading researcher in the field, and he or she said it was not good enough. Okay? But this is what we get. If you kill the animals immediately after the state, in the control case, there's no change, but in the exposed case, there's a difference between slow wave and REM. This is significant. Probably means that this decrease here is significant would be significant if we added more animals, but it means that REM certainly has more phosphorylation of CAM kinase 2 than slow wave, and you say, well, maybe it's those animals. Those animals have more phosphorylation of kinases and more gene expression and more everything. But you do it for ZIF. Again, ZIF takes time to peak. So when you kill the animals immediately, you don't see changes in ZIF. So ZIF in this case is a negative control, because there was no time elapsed so that ZIF could show a change. Okay? So what is very interesting is what is the relationship between this phosphorylation of the kinase and spindles, where we're talking about oscillations in the cortex that are related to learning during sleep. So if you record spindles, here you have a little, the raw data, then you expand it here, you see spindles, you can filter them, same thing here. In the slow wave animal, in the REM animal from different groups. These are state maps that show where you can find the states, not very important. What is very important is here. There is if you look in the slow wave sleep group, animals that were not allowed to have REM sleep. So they have spindles only at the end of slow wave, but they don't trend. They have spindles here, those blue marks here, but they don't have nothing here because they don't have REM sleep. This is the Rem Sleep Cluster. So when you look at those spindles here, they're not correlated with the phosphorylation of CAM kinase 2. Okay? But if you get the REM group and you get those spindles at the transition here, if you add the spindles in slow wave and in the transition, which is the black, you get a very nice correlation, an R square of 0.48, and it's significant. But if you get the spindles only of IS or only of slow wave, the relation is not there. So it's not one state or the other state, it's the spindles. Well, I mean, we think it is the spindles. We think it's all those oscillations in the transition that are proportional to the phosphorylation of chem kinase 2. The work that I'm not going to show to you guys is the modeling of that. The work of Wilfredo Blanco. I actually asked him to come today. He couldn't because he was in a thesis defense, in which we basically modeled that and showed that without LTP, the synaptic weights will wander up and down more or less like Tononi and Cirelli say. But if you add LTP to the system, if you model LTP to the system, you will have changes that are non-trivial that will amount to changes in the Spearman correlation with the original pattern. I know this is crazy too fast, but what it means is that when you add LTP to the system, the synaptic pattern suffers a restructuring. Okay? It suffers a restructuring. The relative rank changes. A synapse that was in the Tononi world, this is the Tononi world here. You start with all sorts of synaptic weights and it all converges to the center. It's a normalization, the dynamic range collapses. Okay? With real data, you still have that. During waking, all the synaptic weights go up. During sleep, they come down. This is the Tononi world with real data. We input real spikes into the network of artificial neurons. But if you add LTP, then you get this restructuring of the trace. Okay, I'll finish with that. Without LTP, we start with a uniform distribution of weights, and you erase it over time. It's good for forgetting. 0.65 is the Spearman correlation of this pattern to this pattern. If you add LTP in two different flavors, the Sejnowski flavor and the Ribeiro flavor, you will get less Spearman correlations, smaller Spearman correlations, and in the case of our model, you come to half of that. So this pattern is, the ranks here are very different. Oh, I think this is the end. Thanks, guys.